Bom dia a todos. Hoje a gente tem um palestrante especial de Toronto, do Canadá, e eu vou apresentar ele. Então agora eu vou trocar para o inglês. Hello everyone, my name is Joyce. I am a PhD student at UFLA, and today I will be presenting a really good friend of mine to our webinar. I would like to introduce Matt Stata. Thank you, Matt, for accepting the NEF invitation. Matt, he's a great plant biologist, and he's a PhD candidate at University of Toronto, and he works with ecology and evolutionary biology, and his expertise is on C4 evolution. During the next 30 to 40 minutes, he will be talking about some convergence and divergence in the path of C4 photosynthesis, aiming to answer some questions about how independent is the evolution of C4 pathway and how many ways it could be achieved, presenting his research with Blepharis. Matt, the audience is yours.
Hi, thanks for the introduction, Joyce. Can you see and hear me okay now? I assume that's a yes.
Yes, yes,
Okay.
I can hear you.
And can we, my slides are up now. Perfect.
Yes,
okay.
Good.
Okay, thanks again for the introduction, Joyce. As Joyce just said, I'm finishing up a PhD at the University of Toronto studying the evolution of C4 photosynthesis. My primary study group is the genus Blepharis in the family Acanthaceae. It's a group of herbaceous to subshrub or some shrubs growing throughout Africa and the Middle East. And we think it's an excellent system for studying C4 evolution because it has a very large number of intermediates as well as several independent transitions to full C4. And so I'm going to tell you today some stories of convergent evolution and what convergent evolution tells us about the different ways to accomplish a particular overarching phenotype, such as C4 photosynthesis. Both on a sort of a grand scale with respect to all the C4 lineages that we know, and in the microcosm of just looking at these elements of convergence and divergence just within one genus, Blepharis. So as this is a talk about convergent evolution, I felt it would be appropriate to start with some words from Simon Conway Morris, who's spent a lot of time thinking about convergent evolution and its implications. And he says, the underlying reason for convergence seems to be that all organisms are under constant scrutiny of natural selection and are also subject to the constraints of the physical and chemical factors that severely limit the action of all inhabitants of the biosphere. Put simply, convergence shows that in a real world, not all things are possible. So in a nutshell, convergent evolution tells us what can evolve, what's easy to evolve, what's difficult to evolve, and the different ways of achieving something. So our story of convergent evolution begins with the enzyme Rubisco, which is, of course, the central enzyme of carbon fixation and the route via which virtually all carbon and energy enters the biosphere. Rubisco, of course, catalyzes the reaction of photosynthesis. But despite its central role in this extremely important and extremely ancient process, it's actually a very slow enzyme. Typically, enzymes of central metabolism have turnover rates of around 80 reactions per second, but Rubisco is about 1 to 10 reactions per second. The other problem with Rubisco is, of course, that it's clumsy and prone to a wasteful, non-adaptive oxygenation event whereby O2 rather than CO2 is fixed, leading to products that the plant has no use for and needs to run a costly metabolic cycle known as photorespiration to deal with. So there's two issues with this dual catalytic nature of Rubisco. One is that you have competition for active sites. So every oxygenation event is a wasted opportunity for carboxylation because it represents an active site of Rubisco that's not doing its proper job. But on top of that, you also have these ATP costs to photorespiration as well. So what's a potential solution to this? Well, here's the Calvin cycle, which I imagine everybody's probably fairly familiar with in this audience. So one solution to this problem would simply be to evolve a better Rubisco. Evolution could come up with a Rubisco that's faster and or not prone to this wasteful side reaction of O2 fixation. But this has never evolved. And the thinking for this is that there's actually a sort of a biophysical limitation to Rubisco here with respect to the ability to discriminate between O2 and CO2, because both of these molecules are relatively featureless. They lack a strong permanent dipole. So from Rubisco's perspective, it's very difficult to distinguish between the two. And that's illustrated rather nicely in this meme that I found online, because yes, people do make memes about photosynthesis and photorespiration. So you can see here is Rubisco unable to tell the difference between O2 and CO2. They're the same picture. Another potential solution would be to evolve sort of an alternate Calvin cycle that uses a different enzyme for fixing CO2. And potentially there's lots of carboxylase enzymes that exist for this purpose, including the one that's been co-opted for C4, PEP carboxylase. So if an enzyme that was able to fix CO2, or probably not CO2, but rather bicarbonate, which is the charged form, if something like that could evolve to be hooked into the Calvin cycle in place of Rubisco, then obviously these problems would be resolved. But this has never evolved either. In the early days of C4 research, it was thought that something like this was going on because the detectable activity of Rubisco in a lot of C4 species was very, very low. But it turned out that that was technical issues with grinding up the very tough bundle sheath cells rather than an actual reflection of reduced Rubisco activity to the point where an alternate carboxylase was needed. Another potential solution that we could imagine is comes in the form of alternate photorespiratory pathways, and these are 5 different photorespiratory pathways that synthetic biologists have dreamed up. That are, in the case of the 4 highlighted in yellow here, they're carbon neutral. They don't result in the release of previously fixed CO2 the way photorespiration does. And in this case of this blue one here, it's actually carbon consuming. So you can see a bicarbonate is consumed here. So this would actually be a net gain of CO2 via photorespiration if something like this could evolve. But despite the fact that synthetic biologists believe that all of these things are relatively promising, none of these have evolved either. And it could be that plants lack some of these enzymes, but if these enzymes have evolved in other systems, why over hundreds of millions of years could they not evolve to fill a similar need in plants? And actually, just as a side note, this photorespiratory shunt right here involves 2 enzymes that the authors had to engineer because they didn't exist anywhere in nature, never mind in plants. So kind of cool, but none of these things have ever evolved in nature. What nature has produced, on the other hand, just a quick summary of what nature has not produced: a better oxygenase-free Rubisco, an alternative to the Calvin cycle with a different carboxylase, or an alternative photorespiratory pathway. None of these have ever evolved naturally. And so far, none of these have successfully been implemented by biologists either. On the other hand, what nature has produced over and over again is what we call carbon concentrating mechanisms, including C4 photosynthesis. And I won't go into any other CCMs here, but there are some interesting ones in algae and other plants, of course. So what is C4 photosynthesis? Well, let's start with C3 photosynthesis, which I'm sure everybody is familiar with, so I won't go over this in too much detail. But you basically have Rubisco sitting in a chloroplast, freely subject to the diffusion of CO2 and O2 into the cell and into the chloroplast, and just at the mercy of the ambient concentrations of these 2 compounds. The carboxylase reaction goes on to produce carbohydrates via the photosynthetic carbon reduction or Calvin cycle. And the oxygenase reaction, fixing of O2, results in the formation of phosphoglycolate, which leads, of course, to photorespiration. C4 photosynthesis solves this problem by restricting the expression of Rubisco just to the bundle sheath cells, which you can see here. This is a mesophyll cell up top and a bundle sheath cell down below, and activating a series of biochemical steps whereby CO2 is converted to bicarbonate by carbonic anhydrase, fixed by PEP carboxylase, and transferred into the bundle sheath in the form of 4-carbon acids, malate or aspartate. And then released, CO2 is then released in the bundle sheath cells by a decarboxylase, and there's 3 different types of enzymes that have been co-opted to do this, which I'll come back to in a few slides. CO2 is released, CO2 levels in the bundle sheath cells are elevated well above ambient, and Rubisco is able to chug along at near saturating conditions with respect to CO2. And then pyruvate or alanine are shunted back to the mesophyll cells, and PEP is regenerated by PPDK to start the cycle all over again. A necessary step along the way from this state to this state is thought to be what we call C2 photosynthesis. And what happens in a C2 plant, it's very similar to what's going on in a C3, with the notable exception that glycine decarboxylase in mitochondria is only found in the bundle sheath cells. So this forces photorespiratory glycine generated from phosphoglycolate to diffuse into the bundle sheath, where it's then released by GDC in the mitochondria. And in this less leaky compartment, the majority of that CO2 that's released by photorespiration can be scavenged up and refixed by Rubisco. So you see Rubisco is still in both cell types, but this bundle sheath Rubisco has the added benefit of all of this CO2 coming in via photorespiration. And if the flux through the photorespiratory pathway is high, this can actually result in elevated CO2 levels in the bundle sheath, such that Rubisco is operating in better conditions in the bundle sheath than it is in the mesophyll. So this is thought to be a necessary step along the way to C4, because in every lineage that we've found where there's something other than C3 and C4 branching phylogenetically between the two, what they're doing is essentially this. And so as a result of this, we've got a stepwise evolutionary model represented here in this pyramid, and you can see this photorespiratory CO2 pump, the C2 pathway right here in the middle as an essential step. So basically you get a series of sort of preconditioning steps, photosynthetic engagement of the bundle sheath cells and enlargement, more chloroplasts, more mitochondria. The mitochondria relocalize to the interior of the bundle sheath before expression of GDC is restricted there. Then you have the establishment of C2. And then from there, you have the upregulation of tissue-specific expression of the C4 enzymes to give you a C4 cycle. But one problem with this model is that we don't really have many extant intermediates that represent the steps between C2 and C4. And there's quite a bit going on there. There's tissue-specific upregulation of a number of enzymes and transporters that have to occur here. And nature hasn't really left us with many intermediates to see how that happens. So I mentioned that C4 is a division of labor between two cell types, and as a result, you get this strikingly different anatomy in C4 plants called Kranz anatomy, from the German word for wreath. So you can see a C3 member of the genus Flavaria here with very uniform green chlorenchyma in cross section. Whereas the C4 species of Flavaria here has these dark green rings around the veins, and that's from the numerous chloroplasts in the bundle sheath cells. And you can see that a little clearer in these light micrographs of two species, a C3 and a C4 of Blepharis, my study system. You can see the large aggregation of chloroplasts in the bundle sheath cells in the C4 that are lacking in the bundle sheath cells of the C3 here. But even with respect to this this basic anatomy of C4, there's divergent ways to achieve this. So in the grasses, which were the first discovered C4 group, there's often two sheath layers mesostome sheath, and a bundle sheath. So you can see here, the mesostome sheath are these tiny ring of cells on the inside, and the bundle sheath cells here surround those with a ring of mesophyll cells around. And in different grass lineages, one or the other of these rings of sheath cells have been co-opted for the function of the bundle sheath, the function of the crayon cells where Rubisco is expressed. So in Stipagrostis, it's the outer ring of sheath cells that have been co-opted. Whereas in Neurachne, the outer bundle sheath cells are just basically functioning as additional mesophyll cells, and the crayon cells are all in the mesostome sheath. And in the grass genus Aristida, things are even weirder. You have two rings of bundle sheath cells, two rings of chlorenchymatous bundle sheath cells, with Rubisco in the interior ring, pepcarboxylase in the mesophyll cells on the outside. And we think what's going on with the outer bundle sheath ring is that those are functioning primarily as for starch storage, as well as potentially refixing CO2 that might be leaking out of the inner bundle sheath ring. And in dicots, the situation's even weirder, with many different types of crayons anatomy known. Most typical C4 dicots use the or express what's called atriplicoid crayons anatomy, where the bundle sheath cells form a continuous ring around the veins with mesophyll cells outside of that. But depending on whether there's water storage tissue of any kind, the situation can get quite different. So you can see these incomplete rings of bundle sheath cells around the vasculature with water storage tissue on the interior. You can see the crayon cells here surrounding the entire body of water storage tissue, and a similar thing going on in the salsinoid and scobarioid anatomies here, with the difference here being another ring of water storage cells on the outside here that's absent over here. And sort of a similar situation in Tecticornia as well, with the bundle sheath cells surrounding the entirety of the water storage tissue. And one of the most interesting divergent ways of achieving photosynthesis, a C4 photosynthesis that any plants have arrived at, comes in the form of what's called the single cell C4 species, Sueda aerolecaspica and Binertia Cynosperceae. And what's going on in these two is the entire C4 pathway, the what would normally be the mesophyll and bundle sheath reactions, are happening just in the mesophyll cells. And in the case of Sueda here, it's an aggregate of chloroplasts in one end of the cell that express Rubisco, whereas these chloroplasts elsewhere in the mesophyll cells do not. And pepcarboxylase out here in the cytosol results in the formation of C4 acids that diffuse into this aggregate of Rubisco-containing chloroplasts. Binertia does things similarly, but a little bit different, with a central clump, a ball of chloroplasts expressing Rubisco, and peripheral chloroplasts that lack Rubisco. And at the level of the whole leaf, you can see what's going on here in Sueda with these clumps of chloroplasts on the interior of the mesophyll cells, and in Binertia, these very, very visible sort of dark gray balls of chloroplasts in each mesophyll cell. So it seems that even something as essential as crayons anatomy is not necessary to achieve C4 photosynthesis. Another aspect of C4 evolution that is divergent between different lineages is the function, as I mentioned earlier, of the decarboxylase, which releases CO2 inside the bundle sheath cells, at least in crayons C4 species. And these are the two most common biochemical subtypes of C4 shown here. By far, the most common is the NADP-ME subgroup. It's the simplest. You can see there's fewer steps here. And then the NADME subgroup involves a few extra steps, and I'll go over why in a moment, and also involves mitochondria, shown here in the bundle sheath cells, whereas the NADP-ME C4 groups only need reactions that occur in the chloroplast. There's no requirement for abundant mitochondria in NADP-ME C4s. So right away, you can see that these steps in both of these C4 pathways are the same. CO2 diffuses into the mesophyll cells, is converted to bicarbonate by carbonic anhydrase, and then is fixed to PEP to produce OAA by PEP carboxylase. But then things get different between these two groups. So you can see here, malate is transported in the case of NADPME C4 plants, and is decarboxylated by NADP malic enzyme to release CO2 right inside of the chloroplasts, which then enters the photosynthetic carbon reduction cycle. In the case of NADME C4s, malate doesn't enter directly, but rather is created within the mitochondria from aspartate that diffuses over and is converted to OAA and then to malate by malate dehydrogenase. And then this has to go out of the out of the mitochondria and into the chloroplast before it can enter the photosynthetic carbon reduction cycle. The reason why different transport metabolites are required here is that when you transport malate in, you are transporting reducing power in with it. And in the case of the NADP-ME-C4s, that reducing power is used to drive the release of CO2 by NADP malic enzyme. And as a result, you can eliminate some of the requirement for the reduction of NADP by the light reactions of photosynthesis. Because the malic enzyme in the NADME is located in the mitochondria, there's not really a readily available way to put that reducing power to use in an NADME C4. Because it's in the wrong compartment. So instead, aspartate is transported in, and you get a cycling of the redox reactions of NAD and NADH in here between the conversion to malate and then the release of CO2. So there's a third subtype as well that uses PEP carboxykinase, but that's only found in the grasses, and it's actually thought to potentially evolve from within one of these other subtypes, so I'm not going to go over that one. It's also much more complicated. C4 photosynthesis has evolved independently upwards of 70 times. I actually think the number is 80 or higher if you assume that reversion is difficult or impossible, and if you assume that C4 lineages don't switch between subtypes because there's not really a good evolutionary mechanism for how that might occur. But what you'll notice about the distribution of C4 plants in this tree of flowering plants is that they're not distributed randomly. You can see there's a cluster of C4 lineages here, and then several clusters over here in the grasses and sedges, with a handful of other lineages all by themselves outside of these sort of hot spots of C4 evolution. And here's Blepharis over here, and Flavaria down here, which has long been a model genus for the study of C4 evolution, although frankly, I think Blepharis is a lot better. So we have established a large living collection of Blepharis at the University of Toronto. Many of our collections come from a 2016 collecting trip to Namibia and South Africa. And you can see several different Blepharis species in their respective habitats here, some of which grow in really, really dry environments that don't see water very often at all. You can see a C4 species here. And what we call a C4-like species here. And I'll get into what that means in a couple of slides. So we believe Blepharis is a potentially powerful study system for C4 evolution because it probably has the largest number of extant intermediates of any lineage known. And critically, the intermediate phenotypes span most of the steps in our stepwise model, including those later steps from C2 to C4. And there seems to be 4 to 5 distinct C4 branches in the tree, at least with respect to what we have in live collection. And one of these C4 lineages within Blepharis has arrived at a very divergent way of achieving C4 photosynthesis with respect to the others, and even with respect to what we know from other groups as well. So it's really kind of cool that Blepharis seems to represent sort of a microcosm of all the things that I find most fascinating about C4 photosynthesis as a whole with respect to convergence and divergent ways of achieving the same thing in different evolutionary lineages. This map here shows where our collections all come from. You can see the red points here represent that 2016 collecting trip, and the blue points scattered either in here or elsewhere represent stuff that's been collected by others and sent to me, or grown from seeds from herbarium collections. And on this slide, you can see the bundle sheath anatomy via transmission electron microscopy of 9 different species of Blepharis that really show this evolutionary gradient from C3 to C4. So you can see a range of bundle sheath ultrastructure and organelle investment patterns here. We can see, first of all, an enrichment of bundle sheath chloroplasts and mitochondria that we kind of refer to as sort of a primed C3. So from this sort of fully C3 species that's outside of the main clade within the genus that's evolved C4 and intermediate phenotypes, you can see an increase in chloroplasts in a C3 basal branching member of the clade of interest, which has evolved C4. Then you can see in what we call a protocrans, you can see this shift of a lot of these bundle sheath organelles into a centripetal position in the interior of the bundle sheath cells. But this is still physiologically a C3. It hasn't established a C2 pathway yet. So GDC is still expressed everywhere, not just in the bundle sheath. Next, we see a further enrichment of that sort of clump of organelles in the interior of the bundle sheath cells, and more mitochondria when we get to the first of our C2 species here, Blepharis diversaspina. This is the Blepharis furcata here. Is what we call a C4-like species. So it has a fully active C4 cycle, but still retains mesophyll expression of Rubisco. So it's sort of running both pathways simultaneously. And you can see that the anatomy is pretty similar to these C2 species, and there's actually still abundant mitochondria, these sort of smaller black dots that are marked with a white arrow here. There's still abundant mitochondria in the bundle sheath of Blepharis furcata, because there is still flux through photorespiration in the C2 pathway because of mesophyll Rubisco. This is an interesting case where we have 2 members ostensibly of the same species, one of which is sort of a special kind of C2 with a low CO2 compensation point and relatively high expression of the C4 cycle biochemistry. And then we also have fully C4 members of this species. So it seems that there might, within Blepharis, there might be a species complex that actually spans that gap from C2 to C4 that I highlighted earlier. And then we see sort of a fully C4, well-derived C4 species here in Blepharis linearifolia. And you'll note that by the time these fully C4 states have been realized, there's very few mitochondria in the bundle sheath cells. And this is typical of the NADPME pathway, where the malic enzyme is localized in the chloroplasts, and there's no need for abundant mitochondria in the bundle sheath cells. So I'm going to do something a little bit unusual and start with the principal component analysis and then go through the data that was used to derive it. I know people usually use these kind of slides as a summary of all of their data, but I'd like to just show, first of all, the clustering pattern that the PCA, based on all of this physiological data, shows. And so here is our cluster of C3 species, our cluster of C2 species, and our cluster of C4 species over here in purple. The C4-like plants, these are 3 different populations of Blepharis furcata, form a distinct cluster right here. And that unusual member of Blepharis mitrata, which is not fully C4, forms a sort of a singleton cluster up here. And then Blepharis gazensis, which is a C4 species, but represents a distinct branch in the tree, as I'll show you when I get to the phylogeny, comes out as another singleton cluster here as well. And this is that lineage that I mentioned earlier that sort of has arrived at a weird way of doing C4 photosynthesis relative to all of these other C4 taxa within Blepharis. So here is part of the data that was used to derive this. This is all of the photosynthetic gas exchange data. And right away, before I go into the details here, I'd just like to point out that you can see that in all of these cases, the purple dots representing the C4 species sort of behave similarly. There's no really obvious outliers here with respect to the photosynthetic gas exchange data. And I will show you in a couple of slides what is driving that difference in Blepharis gazensis. But right here, you can see that from a physiological standpoint, with respect to gas exchange data, Blepharis gazensis is not behaving in an atypical way. So it's all the biochemistry that's driving that separation of Blepharis gazensis from everybody else. But what you'll notice here is we've got carbon isotope ratio, delta 13C, in the x-axis, which is a measure of C4 cycle strength activity. And only when you get significant net fixation of CO2 by PEP carboxylase do you get a shift in delta 13C. And you can see that the C4-like members of Blepharis furcata are distinct but approaching the carbon isotope values of the C4 species. And then in the y-axis here, we've got the photosynthetic, compensation point. So you can see the C3 species have a typical compensation point of around 50 to 60. The C2 species have reduced that substantially to about 10 to 20 ppm. And this one odd species of Blepharis mitrata has reduced it even further, but without any C4 cycle engagement that would shift the carbon isotope value. Here you can see the net photosynthetic assimilation rate at saturating CO2 divided by the rate at ambient CO2. So this is a measure of how quickly the photosynthetic response to CO2 saturates. And you can see the C4 species have flattened out quite a bit, whereas the C3 and C2 do not. They continue to rise when you raise them to saturating CO2. And the C4-like populations of B. furcata are behaving in a somewhat intermediate manner. This is normalized carboxylation efficiency, which is derived from the initial slope of the ACI curve. And you can see that the C4 and C4-like species have dramatically elevated carboxylation efficiency relative to everybody else here. The CI-CA ratio, the ratio of intracellular CO2 over ambient CO2 outside of the leaf, is lower in our C4 taxa as expected and intermediate in our C4-like species. So this again highlights the fact that Blepharis contains species that span that gap from C2 to C4. Stomatal conductance, lower in the C4 and C4-like species. This is a little bit odd. This is really high here and indicates that this population of Blepharis mitrata, this C2 population of Blepharis mitrata, may be suffering from some kind of stomatal dysregulation, the fact that it's got its stomates open so much higher than everybody else. And here we have PEP carboxylase activity. So again, you can see the PEP carboxylase activity largely mirrors normalized carboxylation efficiency, which is what we expect to occur when you start to get net fixation of CO2 by PEP-CAs. So before I get into what makes Blepharis gazensis, that sort of oddball C4 lineage, so interesting, I want to talk a little bit about this population of Blepharis mitrata, as well as what's going on with the C4-like Blepharis furcata. So previously, we did a bunch of isotope screening from museum material. And based on 2 or 3 samples, both B. furcata and B. macro were identified as being C4-like because they had intermediate carbon isotope values of around -16 to -18. we've now done broader sampling across these species, and as you can see, Blepharis furcata sort of spans from a C4 isotope value up to the low end of a C3 value at about -20. B. macra, most of these populations are in the C3 or C2 range, so it seems that most of this species is not C4-like, or at best is very weakly expressing an active C4 cycle that's resulting in net carbon gain. And then really interestingly, these numerous populations of Blepharis mitrata, most of these are down here in the -14 to -15 range, typical of a C4. But there's a handful of populations of Blepharis mitrata that have non-C4 isotope values. These all occur in the southern end of the species distribution down here. But it's important to note that even though this map makes it look like the species range is disjunct with a gap here, there actually have been collections made in here. I just was unable to obtain samples from any of those. And some of these points you see down here have C4 isotope values. So it seems that there may be sort of a species complex going on here with different populations, different genotypes of Blepharis mitrata, either being C2 or C4. And in the case of the one that we have in live collection, while it seems to be mostly C2, it's got enough differences going on that it's been separated out as its own cluster by the principal component analysis. So just to sort of highlight again how Blepharis spans this gap from C3 to C4, I'd like to just quickly present the individual ACI curve data for my collection. So here we can see all of the C3 species, and they all sort of converge with compensation points down here of around 50 to 60. And they also all continue to rise as CO2 increases. And I should mention that these are normalized ACI curves, so these have all been divided by the maximum assimilation rate to put them on a scale of 0 to 1, just to highlight the differences in the shape, irrespective of differences in absolute photosynthetic rate. Here we can see the C2 species superimposed over top, and you can see they mostly behave like the C3 species. They continue to rise as CO2 rises, but the compensation point has been depressed. The compensation point is now around 10 to 20. And that's the result of the ability to refix this photorespired CO2 that's forced in the form of glycine into the bundle sheath cells. Then we see the C4 species that are behaving very differently. A much steeper initial rise, a lower compensation point close to 0, and then they saturate at around 200 ppm intracellular CO2. But then really critically, these three populations of Blepharis furcata kind of fill in this gap in the physiological ACI space. So you can see that they have compensation points similar to the C4 species. They rise sharper than the C2 and C3 species, but not as sharp as the C4, resulting in lower measured carboxylation efficiency. And they don't saturate as early. This is critical. The fact that they don't saturate as early is the result of that mesophyll rubisco. So you have to increase the intracellular CO2 until that mesophyll rubisco is saturated before the response curve saturates. So allow me to shift gears back to explaining Blepharis gazensis and why it's sort of an odd member of this genus. So this plot here that we see is similar to the gas exchange data, but in this case, this is all biochemical data. So this is total leaf activity of 7 different enzymes: PEP carboxylase in all of the x-axes, and different enzymes in the y-axes. And you'll notice that in most of these cases, the C3 and C2 species sort of cluster down here with low activity of both enzymes. except in the case of Rubisco, where they're actually clustered sort of in the top left corner with higher activity of Rubisco, but lower activity of Pepsiase. While the C4 and C4-like species form a sort of a scatter shotgun blast up here with much higher activity of both of the enzymes that are being plotted against one another. But note here that Blepharis gazensis is this outlier in each of these top three plots with very, very high activity of both of the aminotransferases, the aspartate and alanine aminotransferase, and the malate dehydrogenase. So these are enzymes associated with the sort of evolutionary choice of transport metabolite. And while all of these species are NADPME C4s, they all have elevated activity of NADPME and virtually undetectable activity of the other malic enzyme, the NAD malic enzyme in mitochondria. They seem to be sort of using a mix of malate and aspartate. And this is actually kind of typical of a lot of NADPME C4 dicots, that you get elevated activity of both of these aminotransferases as well as the malate dehydrogenase. But the extremely high activities of these three enzymes in Blepharis gazensis suggests that something unusual is going on in this species. And these three aspects, these three data points here are the primary drivers of B. gazensis coming out separately in the PCA that I showed at the outset here. When we look at the phylogenetic tree, the species tree shown here on the left, which is based on 500 genes, we can see that there are, as I mentioned, there are 4 to 5 independent C4 lineages within Blepharis. So you can see this is sort of the main C4 group that's radiated up from Southern Africa into Northern Africa and the Middle East. Here we have Blepharis furcata, which for our intents and purposes we'll treat as a C4 because it has upregulated the C4 cycle. But because it's got a non-C4 species branching in between, we consider it an independent lineage. Same with Blepharis mitrata. There are 2 C4 members of this group, and then that one C2 member here that branches in between, making this an independent lineage. Blepharis seriolata and aspera come out as another distinct lineage down here. Although in some of these phylogenies, which I've generated previously, these 2 species come out basal within the main C4 radiation here, which is why I say 4 to 5, because it's not clear whether this is the correct placement. It's got high bootstrap support of 92 here, but almost every other node in this tree that without bootstrap values shown have absolute 100% support. And then Blepharis gazensis is way down here, very distinct phylogenetically from the rest. And one thing that's really interesting is when we look at this gene tree for the C4 isoform of pepcarboxylase, the gene encoding the copy of pepcarboxylase that's been co-opted for C4 function, we see that all but gazensis form a clade together with no non-C4 species within this clade. And what that means is that with respect to all of these C4 lineages up here in this part of the tree, the C4 copy of pepcarboxylase evolved only once and then was passed around, presumably by hybridization and introgression, between these different C4 lineages up here. And this has been observed in other groups as well, but it's really kind of striking that all of these lineages in relatively close phylogenetic proximity to one another share a pepcarboxylase gene that's evolved C4 kinetics and C4 expression only once in a common ancestor, and then sort of been swapped around. And it sort of seems as though this introgression of a C4 Pepsiase gene has really facilitated C4 evolution within this group. And then Blepharis gazensis comes out down here with its own isoform of C4 pepcarboxylase that has evolved de novo without this facilitation. So this sort of casts an interesting perspective on, are these really independent branches? They're independent phylogenetically because they have non-C4 species that come up and branch in between, but they're clearly they're not evolving C4 completely independently if they're being facilitated by introgression of a pre-adapted C4 gene encoding PEP carboxylase. My aim with future work with this transcriptome data is to characterize exactly how many other genes show similar patterns to this, and whether it's just PEP carboxylase that's been introgressed or whether other critically important genes have been passed around too, and to sort of create a metric for how independent versus how facilitated these independent C4 lineages have been. But the thing that I'll talk about for the rest of this this presentation is how Blepharis gazensis has done things differently. So not only did it evolve its own C4 PEP carboxylase gene de novo and independent of the rest of these guys, but it's got unusual biochemistry with respect to those three enzymes that I showed you. And so I'd just like to call your attention back to the biochemical pathways here, and speak for a moment about the ultrastructure and bundle sheath anatomy of the two and the consequences of the evolutionary selection of one decarboxylase over the other. So I mentioned earlier that the NADP-MeC4 species don't have a need for mitochondria in the bundle sheath cells, whereas the NAD-ME species do, because their malic enzyme is localized within the mitochondria. So right away, we can expect to see a difference between an NADP-MeC4 and an NAD-MeC4 with respect to how many mitochondria there are in the bundle sheath cells. I also mentioned that in the case of NADP-MeC4s, reducing power is transported in the form of malate, and that reducing power is used here. So there's not a need for this reaction to be carried out by the light harvesting mechanisms of photosynthesis. And that has a consequence for chloroplast ultrastructure in NADP-ME versus NAD-MeC4s. I probably don't need to go over this too much, but just a reminder that thylakoid membranes within the chloroplast can be either stromal, where they're just sort of freely interlaced about, or stacked in dense grana. And the dense grana are where photosystem 2 is expressed, which is responsible for that reduction of NADP. So you can imagine that an NADP-MeC4, if it doesn't need that photosynthetic reduction of NADP, would not need abundant grana in its bundle sheath chloroplasts. And that's exactly what we see in most of the C4 members of Blepharis. So down here in the bottom, you can see two transmission electron micrographs of Blepharis ciliaris, which is typical of all but one of the C4 lineages within Blepharis. Very, very little evidence of stacked grana in the thylakoids of these chloroplasts, and also no or very few mitochondria visible in these bundle sheath cells. In contrast, take a look up here at these EM images of Blepharis gazensis. You can see there is abundant thylakoid grana stacking marked with these white arrows. And you can also see there are numerous mitochondria in here indicated with these asterisks. This kind of bundle sheath anatomy is typically seen in NAD-MeC4 species. And in fact, when I showed my PhD advisor these images, we made a bet on a loser buys the winner a beer on whether or not Blepharis gazensis was going to turn out to be NAD-ME. And I won. It's not NAD-ME. It's as you saw from the biochemical images earlier, or the biochemical plots earlier, it has high activity of NADP-ME and virtually undetectable activity of NAD-ME. So what's going on here? Why does it have this sort of odd anatomy and this extremely high expression of the three the two aminotransferases in the malate dehydrogenase. Well, what I suspect is going on is that you have sort of a hybrid cycle that's an odd mix of these two biochemical subtypes. I believe that aspartate is going into the bundle sheath cells as it would in an NADME-C4, but it's being converted to OAA and then by malate, particularly this step, the conversion to malate. I believe this is happening in the chloroplast, which requires high activity of the NADP-dependent malate dehydrogenase, whereas in an NADME species, it's NAD-dependent malate dehydrogenase in the mitochondria. And then this is being converted back to alanine for the return trip back to the mesophyll cells. This would explain the abundant thylakoid grana, because if you're sending in aspartate, you're not sending in reducing power, which you typically would in an NADP-MeC4. And it would also, I believe, explain the abundant mitochondria. And the reason for that is that it appears the isoform of the alanine aminotransferase that Blepharis gazensis is expressing is a mitochondrial localized ALT. So I believe the abundant mitochondria that we see in the B. gazensis bundle sheath cells are essentially there just for the conversion of pyruvate to alanine for the return trip back to the mesophyll cells. So why might something like this evolve? Well, it's possible that Blepharis gazensis, because it's in a divergent part of the tree, it's possible that these high activities of these three enzymes were sort of an evolutionary precondition that led to B. gazensis evolving this unusual biochemistry. I don't have the data to show the expression levels, but my RNA-seq data preliminary results show that B. gazensis doesn't have a significantly higher expression of these three genes, but it has much higher activity. So it's possible that the ancestor, the C2 ancestor of B. gazensis, had some of these genes or some of these enzymes encoded by genes with different kinetics that lent itself to a certain unusual evolutionary process in the rise of C4 photosynthesis here. That perhaps just the kinetic advantage of these genes in B. gazensis was responsible for the unusual way of doing things here. But that's highly speculative, and I'll need some additional transcriptome analyses to be able to peg down exactly what's going on here. So just to summarize everything in closing, it seems that aspartate is either the sole or at least primary transport metabolite that's entering the bundle sheath in B. gazensis, which is atypical of an NADP-ME species. And because of this, reducing power is not being transferred into the bundle sheath cells. So photosystem 2 is still required for the reduction of NADP. And mitochondria are not required for the release of CO2 as they are in an NADME species, because the malic enzyme is still in the chloroplasts. However, the mitochondria are required for the pyruvate to alanine conversion step for shuttling back to the mesophyll cells. There's no particular obvious disadvantage to this variant biochemistry. So it's unclear why this is such a peculiarity, but nevertheless, this kind of biochemistry and anatomy hasn't really been observed in any C4 lineage before. So even within Blepharis, this represents a sort of unique and divergent way of achieving C4 photosynthesis. So it's possible that this represents a transitional step on the way to NADP-ME C4 biochemistry that hasn't been observed before. But I think it's much more likely that B. gazensis has just become evolutionarily locked into a sort of strange variant C4 biochemistry, and that this represents yet another divergent route to achieving the convergent phenotype of C4 photosynthesis. So with that, I'd just like to make a few acknowledgements. I'd like to thank my PhD advisors and my entire PhD committee. Definitely some key collaborators on this work, Jingwang Zhu and Amy Liu at the Shanghai Institute of Plant Physiology and Ecology, and too many collectors and herbarium curators to count who've been able to provide me with seeds or samples of material. But particularly, I'd like to thank Ton Ralkins, who collected the seed of Blepharis gazensis in Mozambique for me. And I neglected to mention this earlier, but it's kind of cool. B. gazensis has only been collected once before in the entire record of all the herbarium collections in 1969 in Mozambique. And I'm a member of a Flora of Tropical Africa Facebook group, and actually made contact with this fellow on there who, within about a week of me asking for it, was able to go out and find a live collection of it and get seeds. So the things you can do with the internet. And of course, I'd like to thank all of my Sage Lab friends and coworkers, past and present, and all of you for your attention for this talk, and just a couple of noteworthy mentions of sources of funding and computing power down here. And so with that, I'll take any questions, and thank you again for your attention.
Thank you, Matt. It was a wonderful talk, and we already have some questions, so I'm gonna read it for you, and I guess you can see it as well. So the first question from Mateus, he's asking, what aspect of our characteristics, the arrangement of chloroplasts, the preference of some groups for specific Decarboxylase enzymes or others, the C4 evolution, do you think were more determinant for its adaptation regarding the increase of CO2 environments?
So if I understand the question correctly, you're asking why do some lineages go one route and some go the other with respect to the subtype? Is that a correct interpretation, do you think?
Yeah, he's asking about the characteristics that you think were more determinant for adapting.
That is an open question in C4 evolutionary research, why you go one subtype versus the other. My PhD advisor has an interesting hypothesis that I won't go into here because he probably wouldn't want me to give away his ideas yet until they're ready to be published. But we don't really know. The NADPME is most common. NADME is the next most common. And the PCK that I didn't talk about is significantly rarer and seems to just be present in grasses. And interestingly, they seem to be nested within other groups. And PCK seems to also require the co-function of NADME. So that's an odd one. With respect to NADP versus NAD malic enzyme subtypes, I don't honestly know why some lineages go one way and some go the other. It could be just by chance of which enzymes have highest expression in the ancestors, or which enzymes have best kinetics. Cleame is an interesting group. It has 3 independent C4 lineages, and they've all gone NADME for some reason. So it seems that group has something that predisposes it to one subtype, to the less common subtype. And then there's a couple of groups that have closely related C4 lineages that are one subtype or the other. So Acanthaceae has 2 C4 lineages, one NADP, one NAD. Portulaca has, I think, 3 independent lineages, one NAD, 2 NADP. And the Sesuvioides tribe in the Azoaceae family has something like 6 different lineages, and they're kind of 50-50 NAD versus NADP. So that would be an excellent system to look at why you go one way versus the other. And we're working to sequence a whole bunch of those right now on another project, which is resulting in why I'm still not done my PhD.
That's good. But I think he's also wondering, like, what do you think in the characteristics of C4 plants? What would like the most important for adaptation, more determinant for the adaptation of plants in the C4 context? Maybe anatomy or biochemistry or like the
With respect to
organelles.
does biochemistry or anatomy help to determine which subtype evolves?
No, just in general, I think. In general, the C4 plants, their characteristics.
Oh, I mean the C4 plants,
I
the physiology shows remarkably little difference. There's some research that says that light use efficiency differs a little bit between different biochemical subtypes, but with respect to the different leaf anatomies and so forth, there's the ACI response curves are almost always the same. The compensation point is close to zero. Saturation occurs at around 200 ppm CI. So the overarching physiology, the overall phenotype, is remarkably consistent despite these underlying, very large, in some cases, divergences of how that phenotype is achieved.
Yeah. Okay, thank you. So we have another question from João Paulo Penac. How do you see the efforts to induce C4 photosynthesis in C3 crops and the potential of that strategy?
I think it's going to be a pretty monumental undertaking, because I think that I think there's a lot of optimization steps that need to occur. We might be able to achieve a transgenic C4 rice or something like that, with a functional C4 biochemical cycle going on. I think knocking out mesophyll rubisco may be potentially a bit of a challenge. And so we might be able to get a sort of a C4-like rice more easily than a C4 rice. But even so, I think it would probably be, it would probably not be as efficient. It would almost certainly not be as efficient as a naturally evolved C4, because I think that there's a lot of optimization steps that go on to sort of get things fine-tuned once you've got a C4 cycle active. And that's one of the things that we've published on before is this reduction in mesophyll chloroplast investment. So one of the things that we had consistently observed for years, but no one had really talked about in the literature, was the fact that C4 species have remarkably fewer mesophyll chloroplasts than their C3 relatives. And so how essential is an optimization like that going to be for efficient C4 function? And how do we achieve that? So it's certainly much more complicated than just introducing all the enzymes in the right compartments.
Yeah, I agree with you.
And then the plasmodesmata requirement is a whole other story as well.
Yeah, and as much as we study, we see how things are very complex. We have another question from Daniela Perez. She's asking, in your opinion, is there a reason why we commonly study Crohn's anatomy for C4, but don't see on textbooks these other arrangements, such as those you have shown us today?
Right. Crohn's anatomy is by far the most common, whether it's sort of the typical atropicoid ring of bundle sheath cells or something unusual where the Crohn's cells surround water storage tissue in some way. That is the vast majority of C4 plants all use that. The two single cell lineages, they each evolved just once, and neither of them have speciated. So there's actually, I think Binertia has two closely related, or maybe three species, but they haven't diversified the way the Crohn's lineages have. So I think for the purposes of a textbook, if you're just sort of introducing C4 photosynthesis at a sort of high level, to just kind of say, hey, this interesting phenomena exists, then it makes sense to just cover the crayons aspect of it. And I know when we teach undergrad courses that include C4 photosynthesis, we don't even go into the different metabolic subtypes, really. We maybe mention it in passing if a student asks a question about it, but we don't go into great detail about NADP, NAD, and PCK.
That's a good point. I personally have my own question.
Okay.
Talking about evolution and trees, phylogeny, do you know how long or like where in the time your C2 and C4 like plants came up, just to see if they appear first, then your C4s, or there is like also a gradient in the C3, C2, and C4 appearance of the species?
So, yeah, I mean, without the benefit of being able to hop in a time machine and go back and sample along a real evolutionary gradient, the best we can come up with is lineages where the phylogeny shows a phylogenetic gradient. So in Flavaria, for instance, the species that the one species that branches most basal seems to be fully C3. Then you get some basal branching protocrans. Then you get weaker C2. Then you get C4. But the issue with Flavaria is that all of the fully C4 species are in one clade, and all of the really interesting intermediates are in another. So it's not a perfect ladder-like gradient. Blepharis, on the other hand, seems to be a little bit more ladder-like with respect to you have basal branching fully C3. You've got a clade that's mostly C2 and protocrans, but then B. gazensis is in there, which is weird. Then you've got a clade with mostly sort of C2 to C4-like and C4, and then you've got the fully C4 main radiation. Another lineage that has a lot of potential for explaining C4 evolution is the grass genus Alloteropsis, specifically Alloteropsis semialata, which seems to have C3, C2, and C4 populations that form a phylogenetic gradient with the C2 clade branching in between. But then other groups are much more difficult to interpret. So, for instance, we had a paper recently on the grass genus Neurachne, and the phylogeny of Neurachne makes it very difficult to interpret. There's not many species, and you've got sort of C3, C4, and C2 branching seemingly randomly. That sort of suggests that maybe you had a sort of an enabled ancestor of some kind, either a protocrans or C2, that sort of, in some cases, spun off species that reverted back to C3, and in other cases spun off lineages that went to C4, but not in a nice stepwise gradient.
Okay, thank you.
And I sorry,
Yeah,
I believe you asked also about dated phylogenies. Was that part of your question?
No, it was just regarding the evolution process,
Okay,
how they appear. Yeah.
Okay.
Thanks. Okay, so I think we don't have any questions anymore, and people are just saying great work. So
Thank you.
Yeah, I think we're done. Thank you again for accepting our invitation. It was a very nice presentation, and yes, I think we're good.
Yeah, and sorry, I seem to have gone maybe 10 minutes over, but I appreciate all the great questions.
Yes, it was a very nice talk. All right.
Okay. Bye, everyone.
Bye, everyone.